Yeah.
So if you want to try to keep track of your own, you can do this one too.
Okay.
Or-
I just
Perfect. All right, thank you.
Okay. Thank you so much. I'm Cari Lewis. I'm a postdoc at Los Alamos National Labs, and I'm going to tell you about a project I've been working on assessing hierarchical population structure in Coccidioides. So population structure refers to the non-random distribution of species in their environment, and there are different ecological and evolutionary factors that can contribute to these patterns of population structure. For example, physical barriers in the environment, such as a mountain range, can prevent migration and subsequent gene flow between two populations. And species that cover a broad geographic region tend to exist, individuals in those populations tend to exist in a much smaller region of that broad population, which we can call substructure. And another way we can think about this is from a hierarchical perspective. At a broad scale, we have those broad geographic regions, and then as we move down through to local areas, sometimes we see specific subpopulations associated with different geographic regions in a finer scale. And the reason why this is very helpful from a disease ecology perspective is it can tell us quite a bit about what's going on in a species and in a pathogen in that host-pathogen relationship. For example, at a broad scale, we can see patterns of admixture and ancestral gene flow, and then moving down to a finer scale, we could identify things such as local adaptation to specific climate factors and environmental situations, as well as in a host situation, a pathogen infecting a host, the social and cultural interactions that might be contributing to these patterns of population structure. And so we all know quite a bit about where Coccidioides is, and so I've compiled just broad geographic structure that has been reported in the literature thus far using whole genome data and plot it in a map. And so we can see that Coccidioides is very consistent in what we see in population structure with substructure in Arizona and a population in the Texas, Mexico, and South America region, as well as in the Caribbean. And then combining that with what we know about Immitis, covers a smaller geographic region, primarily along the western coast and parts of South America. But it has also been reported substructure associated with Washington and parts of California. But the challenge of working with whole genome sequence data in Coccidioides is the majority of the data publicly available is clinical data. But the good news is previous researchers have linked environmental isolates to those clinical isolates, which anchor those genotypes in geographic space. Other challenges that we face in disease ecology is that in disease relationships in those clinical isolates, some of them are travel associated, where they are reported outside of their endemic region, and sometimes there's very poor metadata associated with them. And then some pathogens have multiple methods of migration and reproduction that can complicate those signatures of population structure and also complicate how we might use them with commonly used tools that sometimes require a priori assignment into populations prior to analyzing them. And some species don't always exhibit tree-like structure. And so I think a solution to this, and I hopefully can convince you of this today, is that by applying a data-driven network analysis, we can harvest all of that genomic information from a variety of different samples to identify those patterns of population structure, even with samples that we don't have a lot of good information for. And so the way that, and then we can use this network analysis to then infer those locations of infections as well as where travel-associated cases may have come from and identify those broad to fine scale patterns of population structure. And the way that the network analysis works is that nodes correspond to individuals in the data set. Edges between nodes are proportional to the genetic distance between those two individuals. And when we look at broad scale, which corresponds to many edges, and this is demonstrated in the K-plot in the bottom left, many edges, which identifies those broad scale relationships and patterns of admixture, and then reducing the number of edges allowed between individuals, we can then identify those fine scale patterns of structures. Since a small value of K is fewer edges, it's those stronger relationships such as relatives and clones. And so I wanted to know if I could use this hierarchical network analysis and compare it to what's previously been published to determine if this is a good method in disease ecology systems to identify those locations of infections and the travel-associated cases, as well as finer scale patterns of structure than we've seen thus far in Coccidioides. And so to do this, I compiled whole genome data from NCBI. So I'm really excited for Jason to publish those 700 genomes so I can add that to this study. And so I took that data and applied quality filters. And I can tell you more about this process, but applied quality filters, reference aligned, and used very stringent SNP filters. And then I calculated an identity by state distance matrix, which is what is fed into the network to create that network analysis. And then I also generated a phylogenetic tree, since that's what's most commonly been used to compare relationships between Coccidioides samples. And then I also ran a kinship analysis. And this was particularly important for population genetics work, since a network and population studies are susceptible to overrepresentation in a data set. And so to demonstrate what that looks like, the Excel sheet is my kinship analysis, and the network plot is my initial network of 183 individuals. And the kinship identified groups of individuals that shared over 99% of alleles in the data set, which can lead to overestimation of clusters in the network. And so I reduced those groups down to a single representative of that genotype and reran the network. And so I included this plot to demonstrate why this is important. So the plot on the left is the one that you just saw, and the plot on the right is the reduced data set with 150 individuals, colored by the same original cluster assignment to the 183 individuals, which dramatically changed the relationships inferred among these two species. And so I used the reduced data set to eliminate the overinflation of clusters in these species for the rest of my analyses. And so I'm going to show you today, briefly, 3 different resolutions in the network and the inferences that we can make about it, make from it about Coccidioides and how it compares to what's already been published. And so this is the first network at a K of 50, which again corresponds to many edges and broad scale patterns of structure. Immatus corresponds to square nodes. Possidacea corresponds to circles. And in the network, Immatus groups very tightly together at a broad scale, which is not surprising, and that's what we would expect. But Possidacea suggests it's a much more diverse species, and then we can see, and I just broadly labeled the geographic regions where these samples were from. So if I plot that in geographic space, the same network in geographic space, using published or inferred latitude and longitude. I did not do any spatial analyses. This is just for visualization purposes. Immatus is primarily along the West Coast, and there's a strong Texas, Mexico, and South America group, as has been published in previous studies. And then also a large Arizona group, and those travel-associated cases in the Northeast United States, some of them have reported travel and have been published, and so it does suggest that those are consistent with previously published data. But I was curious about the blue and green clusters in between the Arizona and Texas, Mexico, and South America groups. And so I followed up on those samples and compared them to my phylogenetic tree, where each individual is colored by the assigned cluster. And when I looked at what has previously been published in terms of admixture and studies of population structure at a broad scale, this particular individual at the top reported travel to Texas. And in the paper that published the admixture results, that individual possessed a greater amount of admixture between Arizona and the Texas, Mexico, and South America populations. And so I think it makes sense that these subclusters are grouping in between Arizona and Texas, Mexico, and South America because they represent a more admixed group instead of grouping very closely with each end of the Possidacea network. And so moving to a finer scale of population structure at a K of 20, you can see that the network opens up. And I've, again, broadly labeled just the general location of where these samples were reported. And so things that I noticed is that there are subclusters identified within Coccidioides, as well as within Immitis, there was that group associated with Washington and San Joaquin Valley that completely separated, which suggests strong isolation or some sort of evolutionary or ecological event or influence that's happening that is definitely worth following up on. Similarly, plotting this in geographic space, those travel-associated cases up in the Northeast United States, we can start to identify particular areas of Arizona where they might have been infected. And so then, similarly, comparing these patterns in the network to what has been previously published, the lime green cluster in Coccidioides, those samples have consistently grouped into different clades in previously published data. And so this pattern is consistent among previous work using phylogenetic trees. And then when I dug deeper into the metadata for the Washington samples down in that teal group of Immitis, 8 out of 9 of them had reported travel to parts of California. And so I think it makes sense that they grouped with parts of, into the San Joaquin Valley area and suggest that there might be substructure among those, among that group. And I'm gonna move a little quicker just because I'm running out of time. Okay, and so then at a K of 12, the finest scale structure that I'm gonna show you today, the network opens up even more and you can see that there are a few individuals within Coccidioides that are that connect the Caribbean group of Guatemala and Venezuela, as well as Texas, Mexico, and South America, and Arizona, and even finer scale substructure within Immitis. And then plotting that in geographic space, same idea, the substructure within the Texas and Mexico and South America group, as well as Venezuela and Guatemala are apparent. And then comparing that to what has been previously published. One thing that I noticed was those 5 samples that connect the different clusters of Coccidioides have been consistently reported as basal variants in a phylogenetic tree, which is also consistent with my phylogenetic tree. And I think it makes sense that they would group in this way because then it suggests that those are ancestral variants that then connect these different groups within Coccidioides. And then there's that Nevada group, which I thought was very interesting. I couldn't find if that sample was actually collected in Nevada. It's the lime green one, apologies, from Coccidioides. The other 3 samples in that were associated with travel, although it wasn't confirmed where they had traveled to. And so I think that that suggests also that either in northern Arizona or somewhere in Nevada that there might be substructure among these species. And so I think if we follow up on some of these patterns of substructure with more data, it could be very informative on what's going on ecologically. And so to wrap this all up, I hope that this demonstrates the utility of a hierarchical network analysis in understanding population structure from a data-driven perspective. This doesn't include any geographic information and just relies on patterns in the genome from dense SNP data. And we also identified finer scale population structure that could be informative, but we definitely need to follow up on that because, for example, we could use this network analysis to group samples into populations and then use those populations as, for example, a variable in our modeling, our environmental modeling, to see if that either improves the model or identifies particular areas or variants that maybe are virulent or associated with higher heat tolerance or things like that. But the caveat is these are all clinical samples. It would be very important to include environmental samples in this to see if the network changes at all and if these populations are consistent. And with that, I will thank my project team because this work was not done in a vacuum and they have been give me very good feedback, and my funding sources and Anavive for funding my travel here. And I will take questions.
Phew, I'm glad all my previous work was not wrong completely. It's always satisfying. I think we have time for probably just one question, if anyone has one.
Of course.
Yeah, okay.
I mean, if it's not going to be Royce, then I'll jump up. Really cool, and I really like that. I'm curious about the imminous cluster by itself, which makes sense on the bigger scale, but as we heard earlier, there is admixture between imminous. So are you surprised that that's not showing up in your data?
So you mean, let me go back.
You had just the big red, not
Oh,
this
yes,
slide,
Very
that one.
beginning,
Yeah, yeah, yeah.
uh-huh.
So shouldn't there be some connection between those two groups?
You mean like introgression between Postadacii and immitis?
Yeah.
Okay. Yes. You know, and I've been thinking a lot about that, and I really appreciate you asking that question. So I stopped my network at a K of 50, and so I don't know if I was to extend that, if I would see those patterns of introgression. I did try to run on my SNP data set an introgression analysis, which used a sliding window of 5,000 base pairs and calculated FST. And I actually didn't find any significant introgression. And so it might be because my SNP filters were very stringent, and that was for the network specifically. So
We can talk later.
yeah, yes, absolutely.
All right, thank you so
Okay,
much.
thank you so
Excellent
much.
presentation.